Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Central University's Common Entrance Test question paper for the subject Plant Biotechnology conducted in the year 30th June 2023. If you are new to my channel, please do like, share and subscribe. If you want still previous year's question papers of Plant Biotechnology for CUET PG entrance exam, you can go to my channel Omni Bio Entrance, select the playlist. In the playlist, you search for CUET, in that you will find. So coming to the first question. Etoposide and teniposide, widely used as anti-tumor therapy, are extracted from which plant? So it is Podophyllum hexandrum. Answer is option 3. Which of the following bacteria can actively degrade chitin and cellulose and produce antibiotics such as gentamicin? So it is Micromonospora species. Answer is option 1. Then which of the following is not a sulfur reducing bacteria? All the first three, 1, 2 and 3, they are sulfur reducing bacteria, but Frankia it is not. It is a nitrogen fixing bacteria. Answer is option 4. Then assertion, reason. In many species, the culture medium becomes dark in color in some time. Of course, in certain plant species such as mango, if you do tissue culture, you can see the medium becoming dark in color. This is because of the exudation of phenolics from the cut surface of the explant. So reason, phenolics reach into the medium from the cut surface of explants. These turn dark brown upon oxidation. So both A and R are correct, and R is the correct explanation of A. Answer is option 1. Then next one. Statement 1. Cephalosporins with 7-alpha methoxy group are produced by Streptomyces species, of course, developed to overcome allergic problems associated with penicillins. That's correct. Cephalosporin is a fermented product of Acermonium chrysogenum. That is also correct. So both statement 1 and 2 are true. Answer is option 1. Then protein type and function. To match the following. TUBZ, that is involved in plasmid segregation. A4. MAMK, it is for positioning of magnetosomes. Then FTSEZ, it is for cell division. CRES, it is for curvature induction. So answer here is option 4. A4, B1, C2 and D3. Then next one. Mycobacterial filaments readily fragment into rods and conchoidal bodies because they are aerobic and catalase positive. Yes. They grow very slowly. Yes. Outer membrane is packed with mycolic acid. Yes. Mycobacteria lack evolved porins. Not correct. Mycobacteria do not cross-link with the arabinose and galactose subunits. That is also not correct. So A, B, C is the answer. Answer is option 3. Then plant growth regulator with examples you have to match. Auxin. Auxin is indole acetic acid. A4. A4 is there only in option 1. So option 4. So option 4 is the answer. But we will see. We will read and see the others also. Cytokinin is furfuryl aminopurine. Yes. Gibberellins is GA3C1 and ethylene is ethophon D2. So 4 is the right answer. Then assertion and reason. Certain differentiated cells undergo changes to become meristematic. Yes. This is called dedifferentiation. Of course, dedifferentiation is the process by which the already differentiated cells they convert into meristematic cells. So that is called as dedifferentiation. So A is correct. Renewed and enhanced RNA and protein synthesis occur during dedifferentiation. Yes, of course. And R is the reason of A. So both A and R are correct, and R is the correct explanation of A. Answer is is option 1. Then next one. Which of the following is known as serrated wintergreen plant that obtain most of their carbon compounds from parasitized microbes? That is Orthia senda. Answer is option 2. Alpha proteobacterial genera are able to form nitrogen fixing nodules with legumes. These include Azorhizobium. Answer is option 1. The next one. Secondary compound identified from Phaseolus vulgaris which demonstrates insecticidal properties is. So it is Arkeline. Answer is option 3. The Anamox reaction is an anaerobic reaction performed by. So what is Anamox reaction? It is a microbial process that converts ammonium into nitrogen gas under anaerobic conditions. And out of these options, it is performed by planktomycetes. Answer is option 2. Then denitrification process is demonstrated by which bacteria? So denitrification, it converts nitrate, that is NO3, to nitrogen gas under anaerobic conditions. And out of these options, Geobacter reducens is the answer. Then which of the following is a free living anaerobic bacteria? It is Clostridium species. Answer is option 1. In bacterial nitrogen cycle, which of the following serve as an electron donor? So NH4 plus serve as an electron donor. Answer is option 3. Then sterilization technique with the material sterilized you have to match. So a dry heat 160 degree to 180 degree for 3 hours is empty glassware culture vessels. 2 itself is A3, so that must be the answer. We will see. Autoclave nutrient media glassware B2. Filter sterilization heat labile compounds like GA3, ABA, etc. Wiping with 70% ethanol, it is the platform of laminar airflow. We wipe with 70% ethanol before starting tissue culture experiments. So A2 is the A3, B2, C4 and D1 is the answer. Then next one. Antimicrobial activity cannot be measured by dilution susceptibility test, yes. You can measure disk diffusion test, yes. The E-test. So these 3 methods can be used to test antimicrobial activity, but Ames test is not used because it is used to detect mutagenic properties of a chemical substance. So answer here is option 4. Disease black leg in rapeseed and turnip rape is caused by. It is caused by Formalingum. Answer is option 4. Then next one. The first DNA based molecular marker used for the study of DNA polymorphism in variety of plants was. So it was restriction fragment length polymorphism or RFLP. Answer is option 2. What is the role of plant jasponates in plant-plant interaction? They enhance plant competition. No, they suppress plant competition for resources. Answer is option 2. Then next one. How does gibberellic acid stimulate stem elongation in plants? By promoting cell division and elongation in stem tissues. That itself is the answer. Answer is option 1. In the direct method of protoplast isolation, the enzyme mixture consists of. So only in one option macerozyme is given, but we will read the whole thing. 0.5% macerozyme, that is 3 option. 2% cellulase in 13% sorbitol at pH 5.4. So that's correct. Answer is option 3. The next one. After a period of time, it becomes necessary to transfer organ and tissues to fresh media. That is called as subculturing. This is because. So why do you have to do this? The culture vessel gets damaged. No. That cannot happen. Only if it can happen only if by chance it falls from your hand and it is broken, then it can happen. But that will not happen because the explants are your own plants, your own tissue culture plants. So you will not try to break the culture vessel. So answer A is, option A is not correct. Culture medium is depleted of its nutrients. That is correct. Photo period cannot be maintained. That is always maintained in tissue culture laboratories. Drying of culture medium. Yes, that is also correct. So B and D only. Answer is option 4. Then next one. Nitrogenase enzyme in Frankia species is oxygen insensitive. That is not correct. It is sensitive to oxygen. Statement 2. Frankia species are microaerophilic and can fix atmospheric nitrogen. Statement 2 is true. So statement 1 is false, but statement 2 is true. Answer is option 4. Probably protoplast viability can be tested using, it is by using fluorescein diacetate. In the options, in the answer key it is given some other option, but fluorescein diacetate is used in protoplast viability test. Then which of the following protoplast induced fusion method is applicable for inactivation of Sendai virus in animals? It is NaNO3 treatment. Answer is option 3. What are asymmetric hybrids? Fusion of protoplast isolated from sterile lines? No. Fusion of protoplast isolated from actively dividing tissue of one parent and quiescent tissue of their parent. That's correct. Answer is option 2. Then haploid plants are obtained by the following methods. Placing another or isolated, it is not another, it is a printing mistake, it must be anther, or isolated pollen grains on a suitable culture medium. That will give rise to haploid plants because pollen grains are haploids. Using leaf disc as explant material. Leaf is diploid, so it will not give rise to a haploid plant. Using nodal explants, again diploid. Using endosperm as explant material, that is triploid, so it will give rise to triploid plants, not haploids. Then next one. What happens during the process of de-differentiation? It's already been asked just previous question. The cells of the explant material stop dividing. No. That doesn't happen. De-differentiation is the process by which already differentiated cell revert back to meristematic tissues. That is de-differentiation. So the cells of explant material stop dividing is not correct. Renewal and enhanced RNA and protein synthesis leads to terms meristematic cells from explant. That's correct. 2 is correct. The explant materials turn dark due to the production of secondary metabolites. No. Somatic embryo doesn't have anything to do with the de-differentiation. So 2 is the answer. All the material used for culture work must be fed from microbes. Which of the following approach is used for this? So filter sterilization can be used. Colchicine treatment is for chromosome doubling. Incubation at 37 degrees, it is usually for bacterial culture growth. Wiping with wet cotton cloth, that also will not help in sterilization. So answer is option 3. Then all plant material used for the culture are surface sterilized to avoid any contamination. Which of the following is not used for surface sterilization? So out of these, jasmonic acid is not used for surface sterilization. Answer is option 3. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you are new to my channel, then If you want still previous year question papers of plant biotechnology, you can go to my channel playlist, that is Omni Bio Entrance. That is the name of the channel. You can go to the playlist there. Search for CUCET, and in that you will find previous year question papers of plant biotechnology. Thank you.